Okay, everybody. We're going to get started with the last talk of the day here. And we are very lucky to have our hometown speaker here from CIMeC, Roberto Bottini.
Thank you. I'm going to use this microphone. Do you hear me? Also on the back? Yeah, it's fine. OK, perfect. So thank you. I'm very happy to be here. I am a CAOS aficionado, I think, because since I discovered this conference, I haven't missed a season, basically. And I've been giving a lot of talks lately, but I have to say that giving a talk here gives me different feelings because it's my hometown, it's where I live, it's where I work. It's a very familiar place with a lot of familiar people. So it's like to give a talk to mom and dad, basically, at Christmas. OK, I'm going to start because I know that I'm the man between you and your aperitivo. So I'm going to be quick and start with a little bit of philosophical reference, just to paraphrase Schopenhauer in saying that before Kant, we were in time and space, but now space and time are in us. And Schopenhauer, with that, wanted to highlight one of the main contributions of Kant, which is also one of my favorite philosophers, and the fact that space and time are not out there, but spatial and temporal relationships are in our mind, and like some sort of schema that we project on our experience, that we project on reality. And indeed, space and time are rather fundamental experience of our fundamental dimensions of our experience. We can take, for instance, our life or our career. Every event happens somewhere, and therefore spatial relationship, and somewhen, temporal relationship. Now, discovering the cognitive key that can unlock both the representation of temporal dimension and spatial dimension in our mind would be a big deal for cognitive neuroscience already. Now, imagine that the same representational mechanism can be used to represent any type of relationship along dimensions that we think are very important for our life. There might be something else in life, but these are pretty important here. I mean any type of abstract relational structure, existing or invented for the sake of the task. So what I'm talking about is a mental representation of abstract relationship across any domains of experience, which putatively is the fabric of our thinking, what put everything together and allow us to think. So this mental representation, they say, it's a cognitive map. And if this is true, you can understand why this concept has become so central in the last few years in cognitive neuroscience. The concept of cognitive maps was introduced by Thorman back in the 50s. And shortly after, it was associated with a very specific region of the brain. I'll give you an hint, is the hippocampus. And indeed, we know that the hippocampus is important for declarative knowledge, for declarative memory. We know that from the most famous, probably the most famous patient in cognitive neuropsychology, which is patient HM, who developed a very profound retrograde and anterograde amnesia after the removal of the hippocampus and neighboring region bilaterally. And so cognitive scientists started thinking that the hippocampus is the place where you store all your memories. But if you have to store it, then you better store it with a structure already. So you have to retrieve not only the objects, the events are represented there, but also how they are related with each other. And evidence that structure is in there in the hippocampus appears a few years later, around the 70s, the 80s, from a different line of research, which is electrophysiology in rodents. With the discovery of spatially selective cells in the hippocampal formation. Maybe the most famous one is a place cell. I'm going to show a little video here. So place cells are situated in the hippocampus proper, and these are cells that fire for a specific position in place. You see here the rat is doing a foraging task. It's going around a cage looking for food, usually. And here they are measuring one cell in the hippocampus that's fire in a particular point of the environment. And if we speed up a little bit and we look at the heat map of it, what appears is a place field. Now imagine one place says one position, another place says another position, so on and so forth, and then you can cover with a map all the environment. One synapse is away, in the entorhinal cortex, we have a different type of neurons. These are called grid cells. And I speed up here a little bit. You see at the beginning, it seems like these cells, this is one cell. It seems like it fires everywhere. But actually, that is not the case. If you wait a little bit and then you run some autocorrelation, you will see the typical six-fold or hexagonal structure emerging. So one place cells fire for different positions in space, but these positions are located at the vertex of equilateral triangle, tiling the space with the beautiful hexagonal ring. And after that, a lot of different cells, an entire hippocampal zoo has been discovered, right, with different species. Right? You have object vector cells, you have head direction cells, the signals where you are going allocentrically in the environment, reward cells, where is the reward, the boundary vector cells, boundary cells, and so on and so forth. And therefore, people started thinking that this is what gives or represent the GPS of our brain, so a spatial map in the hippocampal formation. But very soon, we realized that it's much more than a spatial map. And indeed, these cells represent relationship across different type of dimension. So probably the first one discovered was time. So with the discovery of time cells in the hippocampus. Here you see a rat that is doing a little bit of exercise, so he's on a treadmill. Now the treadmill starts, and there are three neurons. The green one that fire at the red one at the beginning of the task, the green one in the middle of the task, and the blue one at the end of the task. These are three different neurons in the hippocampus. And so neurons there, they not only code for a specific position in space, but also for a specific moment in time. And now you see that these neurons are also place cells. For instance, the red neurons fire for that particular intersection in this 8-shaped maze. And if we go on with the video, the other neurons will fire in different positions of the maze when the rats take the other route. A few years later, we had these other very important milestone experiments by Dmitry Aronov. And what rodents was doing here was to learn specific frequencies, musical notes, if you want, like a D diesis, A major. They listened to a sound that was either increasing in tone or decreasing in tone, and they have to release a lever when the sound hit a specific note. Right? It takes a while, but then the rats learns to do that. And then in the hippocampus, you start seeing the place cells. Also, some of the place cells develop a place field for a specific sound frequency. And grid cells develop fields for multiple sound frequencies. So again, the same cognitive machinery is used to represent locations and structure in this case, an auditory unidimensional space. What about humans? Well, in humans, you can measure grid cells and place cells directly, but that is very rare, just in some conditions. But luckily for us, we can measure the activity of grid cells non-invasively using, for instance, fMRI. That was discovered, this method, back in 2010 in this landmark paper. Imagine people are in the fMRI, they navigate their virtual environments, like this ring. And in a foraging task, they go look for different objects in the environment. In doing that, they design trajectories in the environment. Now, these trajectories can be aligned or misaligned with the main diagonal of the hexagon. And aligned trajectory, they go through a larger number of firing fields, and therefore they make the neurons firing more compared to misaligned trajectory. You can detect that at the population level using fMRI, and the results look like this. So a sinusoidal modulation of the bold signal that emerge in the entorhinal cortex, and of course a sinusoid according to a 60-degree modulation with aligned versus misaligned trajectory. And you can do that in control periodicity that are not biologically plausible, like 90-degree or 45-degree instead of 60, and you don't see that. So now you have a way, non-invasively, fMRI, but also MEG, it works, to look for this grid-like coding, and then you can really go with your imagination and ask people to navigate any type, as I was saying before, of relational structure in any dimension, be them more concrete or abstract, but non-spatial dimension. And that has been done over and over, this is now a very replicated results. So in a feature space, you can see this grid-like coding emerging when people navigate an odor space in fMRI. So there is nothing to see there, just smelling odors and different combination of them. Completely auditory space with pitch and duration. A social space in which people are represented and compared in terms of popularity and competence, or a pitch size multidimensional space. This is represented also only with words that are associated with this dimension. And this is work that is done here at CIMeC by Simone Viganò and Manuela Piazza. So a lot of evidence that what are they telling us? They are telling us that cognitive maps provide a low-dimensional mental model to organize knowledge across potentially any domain of experience. If that is the case, then searching for information in memory relies on the same neurocognitive mechanisms that are involved in spatial navigation or spatial foraging. That is an idea that I think first was suggested by 𝗥ន់💧 and Moser. And the work in our lab in the last few years was targeted to test these hypotheses and to try to push the boundaries of this metaphor. To what extent this parallel is correct, when it's going to break at a certain point. To start with, spatial navigation doesn't it happens in across different reference frames during for humans and basically for any animal. An allocentric reference frames, which is the one usually that is associated with cognitive maps in the hippocampus, in which different objects are localized according to the relation with each other or to the relation with external landmarks. Or egocentric reference frame in which we localize objects depending on our vantage points. So if I do that, you are on my right. If I look on the other side, it's different. You are on my left, but you are always east, right? In that case, I think at least. This navigation, spatial navigation across reference frame is supported by a whole network of regions, and two very important regions, and that is a typical dissociation that you find in these experiments, is that allocentric representation associated with the medial temporal lobe. Here we see a very important study with allocentric encoding in the entorhinal cortex, and egocentric representation are traditionally associated with the parietal cortex. And here we see in these experiments in virtual reality, particularly with the precuneus, but we know that also posterior parietal cortex, inferior parietal cortex, they're very important for egocentric representation. So the question is, if we want to push the boundary of this metaphor, is that the case that also our conceptual navigation, mental representation of abstract relationship happens across complementary reference frames? So to figure this out, we designed this molecule experiment, and this is work led by Simone Viganò when he was postdoc in the lab, and now he's at the Max Planck Institute in Leipzig, and he's going to be soon on the job market, and he's a fantastic scientist, so keep your eyes open. Simone told participants to imagine to be scientists in a future society that live in peace and prosperity thanks to the discovery of two particular molecules. one molecule that gives you extra mental strength and a molecule that gives you extra body strength. We call it megamind and megabody. This was just a cover story to have a little bit of fun. Participants were trained to recognize the particular configuration associated with these two molecules. And you see they are virtually identical, except for two things, the color, and the other thing is the length of the upper bond and the lower bond. So in the fMRI, what participants have to do was how I call it, morphing match to sample task. So what does it mean? That the molecule started from a random configuration and then started morphing, right? So the two bonds started shortening or lengthening according to different rates, right? They do that for a second, they stop, and then participants had to keep morphing the molecule in their mind, right, with the same rate, and decide whether, according to that morphing, it will transform in one of these two targets. Depending on the color, megamind of megabody. So this is just like a morphing experiment, if you want, like a morphing task, morphing the shape of the molecule. But we know that geometrically, that can be intended as drawing a trajectory in a two-dimensional space that is defined by the lower bond and the upper bond. These two molecules are two particular points in this two-dimensional space, and you see that they are in the opposite side of this two-dimensional space. And each morphing designs a trajectory in this two-dimensional space. Of course, this is the case if, in order to do this task, this organization, two-dimensional organization, is activated by the subjects. And that is the case. So we ran the typical hexadirectional coding analysis to look for grid-like coding in the entorhinal cortex, as I showed you before. We used RSA in this case, but the principle is the same, aligned or misaligned according to a 60-degree rotational symmetry. And we find this grid-like effect in the left entorhinal cortex and no effect in all the other control condition like 45 degree, 90 degree, 57 degree instead of 60 degree. That was highly expected. What was interesting is that this six-fold symmetry effect, or this grid coding effect, scaled with the distance from the goal. And it was stronger in the quadrant where the goal was present. And this is interesting because the same phenomenon appears when you measure real grid cells in rodents that goes around in an environment. These are the two papers that shows this effect. When you put some food in an enclosure and the rodents learn that that is the place where there is food, then grid cells around that place starts firing with a higher firing rate, even though then you take away the food and there is no reward there. So grid cells are signaling where something interesting is happening in the environment by increasing the firing rates. And that is the case both during spatial navigation and during, let's call it, feature navigation or conceptual navigation in human. But what about this egocentric signature? Okay, we see this allocentric signature in the medial temporal lobe that was highly expected. To test whether there is a similar egocentric representation in the mind, we make so that the different trajectory in the space, so the morphing of the molecule, was of one of 7 different types. Either they were on target, so the morphing molecule will become one of the 2 target molecules, or they were off target for a given number of degrees, like off target 45 degree, 90 degree, or 135 on one side or the other side of the molecule. You can call it left and right. So here, for instance, you see 3 different trajectories that have the same egocentric signature in the sense that are 45 degree on the right offset of the target. You see that these different trajectories allocentrically are different from each other. They point in a different direction, that are in different location of the space. But they are similar egocentric. What does it mean egocentric here? You have to interpret that geometrically in the sense of bearing from egocentric bearing from the direction of motion that constitute, in this case, the vantage point. And this condition, egocentrically, is exactly the same as this condition, even though allocentrically this trajectory has a odd difference and the target is on the other side. So what is the brain region that treat these two trajectories as similar and these two trajectories for these two conditions as different? Simone ran two types of analysis, both, I'm going to go a little bit fast here, both repetition suppression or adaptation, or NVPA, so neural decoding to distinguish the different conditions. And in both cases, we found different regions of the parietal cortex encoding for this egocentric signature. When you go and see which region was activated in both conditions, so showing strong invariance and encoding across different methodology, was this part of the precuneus, which is exactly the same regions which is activated when participants navigate virtual reality and encode the egocentric position of objects. And indeed, you can take this little spots here in the brain, this cluster of activity, and do dimensionality reduction analysis to reduce that from the voxel space to 2 dimensions. And you see that here, the different conditions are represented differently, with the on-target trajectories are here, trajectory on one side are here, trajectory on one side, the other side are here, and with difference also in degree, 45 degrees farther away from 90 and 135 that are the trajectory that are more off targets. So this is to say that we can push a little bit the boundary of this parallel and this metaphor between spatial and conceptual navigation, because during mental exploration, the human brain recruits in parallel both allocentric and egocentric representational mechanism schemes as it happened during spatial navigation. So if we have to, in our conceptual space, try to think what is the car that we need to buy and that we want to buy according to the dimension that we are interested in, that are important for us, there will be grid cells signaling allocentrically where is located this target in our conceptual space, and also a parietal code that tell us how to orient ourselves in this space. Let me just mention here that the similar results, we obtained similar results in a different task in which people were involved in a mental time travel task, which is this ability of humans to self-project themselves either in the future or in the past, and then judge temporal relationship for that new vantage point. So for instance, if I self-project myself in 2015, COVID-19 is in the future. It's no longer in the past as it is if I am where I am now. And a lot of people have suggested that this ability, that it might be, at least to some extent, exquisitely human, is based on the complementary use of an allocentric and an egocentric representation of time. And that's what 𝗥cie𝗯𝗲𝗿𝘁cie shows in this paper that is out. I don't have time to go through that. But that is to say that this complementary use of frame of reference extend across different type of conceptual navigation, if you want, or mental representation of relationship. Okay, but how do we navigate conceptual space? If you want to really push the metaphor a little bit forward again. So when we navigate space, here let's see some OpenAI magic. When we navigate space, we move with our body, right, in the physical space, and we receive a vestibular input, we receive head direction input, we receive velocity input, and all this information that is processed by different regions of the brain arrive to the entorhinal cortex, and that makes so that grid cells and place cells can track exactly the position where we are. But when we mentally, oh, there it comes again. Yeah, but when we mentally navigate our conceptual space, there is nothing of that. What are we doing in that case if we are doing something? In order to answer this question, we have to take a step back from conceptual navigation and consider what happens to these neurocognitive mechanism in the hippocampal formation in primates, because it's slightly different than what happens in rodents. In primates, if you try to find the place cells and grid cells that tells you where the monkey is, right, in a space, you will not find many. You will find some, but not many cells that would do that compared to rodents. Instead, these cells start firing for visual behavior. So place cells become spatial view cells. So one cells will fire for one position in my visual scene, another cells for another position, not in a retinotopic way, right, but really for the position. Grid cells would encode the viewing behavior. So one grid cells will fire when I look here, when I look there, when I look there, when I look there, tiling this visual space with this 60-degree hexagonal shape. And head direction cells becomes saccade cells. So they fire for different angles or direction of my saccade behavior. And that has also been found for grid cells in humans. That was monkey data, right, with a single cell recording, but you can find a grid-like coding for visual behaviors in humans across many tasks, both in fMRI and in MEG, for instance. Now, even more interesting, you don't even have to move your eyes to activate these neural code for visual behavior. In these experiments, the monkey was instructed to fixate, otherwise no reward, and then to follow with the covert attention a dot that was moving on the screen according to a particular trajectory here. And even if there was very minimal movement of the eye, then grid cells tiled anyway this visual system, activated only from the position of covert attention. And we recently showed that that is the case also in human, with grid-like coding activated only with covert attention of movement within the environment using MEG. This is work by Giuliano Giari while he was a PhD student in my lab. I don't have time to go through all these experiments. But what we conclude, Giuliano and I, from this paper and from this study, is that maybe this attention modulated activation of grid coding during can be a suitable mechanism for the activation of the same machinery during the navigation of conceptual spaces, where there is nowhere to go and nothing to see, but in principle you can move your attention and your internal attention in your conceptual space. So we reason that if that is the case, and given the tight relationship between the attentional system and the oculomotor system in the brain, then we should be able to see a behavioral signature of this conceptual navigation. So when people conceptually navigate, let's say, then maybe we can see the structure of what they are thinking about reflected in their spontaneous eye movements. And so how did we test that? So we put people in a dark room, switch off the lights, but then we had infrared camera-based eye tracking, so we can track their eyes. And we asked them to engage in a random word generation task. So they have to say one word after the other, keeping the tempo of a metronome, so more or less one word every second. And we had two conditions. One, there was a replication of a previous studies, a numerical condition, number from 1 to 12. Tell me all the number from 1 to 12, one at a time, but in the most random way as possible. And the other condition was colors, so 12 colors. Tell me the colors in a random way as possible. And this would represent like a one-dimensional space or a two-dimensional map. And then what we did, again, following a previous study, we took the average, we correlated the distance in the conceptual space. So for number, for instance, the distance from 3 and 7 is 4, but from 7 and 8 is only 1, so small or large distance.
Okay, no problem. That is just to switch microphone.
Is it much better? Guys, I'm sorry, I should have told me. I would have switched before. Okay, so you correlate the Euclidean distance. You take the Euclidean distance for each of these fixations, you put it in a matrix, you correlate with the matrix that comes from the Euclidean distance from the ratings of similarity judgments. You do that for each participant, and then the correlation is significant. So it's telling you that some of the structure of this color wheel is maintained also in the spontaneous gaze behavior. And indeed, if then you do an average across subject of these two matrices, and you run multidimensional scaling, you see that for the similarity judgment, as we know, you find this beautiful color wheel. But if you do that on the gaze, you don't find a wheel, but you find some structure. You see all the blues are here, and then there are the greens, so the blues, the greens, and then it goes the reds and orange, and then all the violets together. So some of the structures is maintained there. All right, so after we ran all of these, we were looking at the data, and we noticed something interesting on top of that, and it's this. So independently of the specific position of the gaze for in the mental timeline or the color wheel, every time people was retrieving colors, they were looking in one side of the space. Every time people were retrieving numbers, they were looking in a different part of the space. It was like they were partitioning their visual space with one category here, another category here, then maybe to better retrieve the items within the category. This experiment was not designed to test this hypothesis. So we designed a new experiment with this in mind. Maybe as we organize things in space, in the real space, in order to then be better in retrieving that in your wardrobe, and you have the T-shirts in one place, the trousers in another place, socks in another place, or maybe not, right? But the majority of people do that so that it's easier to get it when you need it. So maybe that happens also for conceptual spaces, in which when we have to deal with different category, we separate this category in our mental space. And this maybe is reflected by spontaneous gaze dynamic. And so Simone and Giulia teamed together to figure that out. The experiment was very easy. So we teach to participants two types of category the day before. Online, just auditory. Category 1, maybe 15 type of jobs. Category 2, 15 different animals. And they have to learn that. And then they came to the lab the day after, and they do our usual looking at nothing word generation task, divided in blocks. Right? So this is, for instance, one run. And then in one run, you say, OK, now tell me all the animals for 40 seconds. And they have to retrieve the animals following the metronome. Now animals again, and then jobs, animals, and so on, like counterbalance so that we can do analysis the data you would see. What happened there? And then, of course, we took the median XY position of the gaze in the 500 milliseconds before they say the word. So what happened there, you can already see that in some exemplar subjects. So they partition their visual field so that every time they were retrieving a category, these are different words belonging to the categories. Every time they were retrieving animal, and so they were looking here, and jobs, they were looking here. And that was idiosyncratic for each participant. Different participants have a different partition of the space, but what was true across them is that they divide their attention in this way. We can, of course, do some statistics to see whether that is true across the sample. So we can take the center of mass of the two categories, we take the distance of it, and we make sure that the between category distance is higher than what it is expected by chance, and that is indeed the case. Or we can do an RSA approach. So we take all the distance in the gaze space from each word that has been pronounced in that particular run, within and across category. So you end up with a distance matrix, a Gaze distance matrix, and then you can correlate this distance matrix with a category model matrix. And then also we did it with a temporal model matrix because we noticed some temporal drift in the gaze. And when you do that, you see that there is an effect both for the categorical and also the temporal. And then you can do also partial correlation to make sure, and that is the case, that the categorical effect is there even when you control for the temporal effect. Now, then Simone and Giulia did the same experiment twice, like again, slightly different, but so for internal replication. I'm not going to show you the data because it's exactly the same. But that is to tell you that then when we put the two experiments together, so we reach like about 60 participants, it's very interesting and for me crucial, a data point emerged. So that the stronger the partitioning, so the more a subject was partitioning the category in his visual/mental space, and the more words it was able to retrieve. So supporting the hypothesis that these gaze dynamics are not epiphenomenal, but are actually useful to do the task that you're doing. In this case, like retrieving items from the category. One of the last things I'm going to tell you, so interestingly, a couple of weeks ago, I think 3 weeks ago, this paper came out in showing what I think is the same effect in monkey. So they reanalyze a lot of studies in the lab, and all studies have the same structure. Was a delayed match to category task, in which monkey was exposed to clouds of dots that had apparent motion in one of 6 directions, basically. And they divided the 6 directions in 2 groups of 3 arbitrarily, saying this is category 1, and actually this is category 0, and this is category 1. And the monkey have to learn what is the categorical boundary to do the task. And the task was the following. So there was a fixation point, so the monkey had to fixate in the middle for all the tasks. Then there was a sample, so apparent motion maybe in this direction. Then a delay, and the monkey was keep fixating in the middle. And then a match that could be of the same category or a different category. And what they did in reanalyzing all this data, they look at the eye movement in this delay. And it was particularly interesting because monkey had to keep fixation there. So they had to look at very microscopic eye movements. And what happened there is that monkey, they were fixating in different position in the space, like within the 2 degrees of visual angle that they were allowed to move the eyes for the fixation, for the different categories. And each monkey, they tested 11 monkeys, every monkey showed the effect, and each one in an idiosyncratic way. Some people partition the categorical space in this way, some other animal monkey partition it in this way. And this partitioning has nothing to do with the actual direction of motion. So they run several controls for that. It really reflects the categorical boundary. This is category A and this is category B for me, even though there was nothing to see. They were not instructed in any way to move their eyes. This is another way to show it, to show this effect with category 0 and category 1. And you see that it's very similar to the effect that I showed you before in our categorical partitioning task. And also in their case, this effect was related to behavior. So if the type of gaze pattern that you find in correct trials here was very different than the pattern that you find in incorrect trials. So when they were looking in the wrong space, according to their categorical partitioning, the monkey was not correct at the end in defining the category. How am I doing with time? Almost at the end, right? Yeah? Okay, like 5 minutes, probably. Okay, then I'm gonna, well, what they conclude, that is a quote for them, is that core spatial and oculomotor brain networks are recruited to mediate abstract cognition. And that's very similar to what I am proposing here. So here I go very quick, but I want to acknowledge also the work of Alex, who is a PhD student in my lab. He did a lot of work. I'm just going to talk of a fraction of his work, but it's also to reiterate this relationship with behavior and to give some hints that these effects are not epiphenomenal. So Alex taught people to modify the mental number line. It's a mental number line in numerical relationship at the end, in which you, from each number, you can go to some number, but you cannot transition to some other number. So for instance, if you are at 22 and you want to go to 21, you cannot just go back. You have to go to 23 and then go to 21, and so on and so forth. They learned that by trial and errors, just looking one number after the other, trying to move and so on. They learned that for different portion of the mental number line, but since this structure basically replicated every 5 items, then they can generalize after once it's learned, you can generalize to other portion of the mental number line. And well, long story short, we look at the eye movements when a number was shown to them, and then they have to think, oh, where I can go from this number, which are the possible transition, which are not the possible transition. And what happened is that subjects look further right for numbers which afford positive action. We call it like that in this context, right? So positive transition compared to numbers that afford negative transition. And then again, the more people was doing that, and the better they were in the task. Then Alex, of course, we did that also in fMRI. We find some interesting result in the entorhinal cortex. But let me directly go to the discussion and to the conclusion, and then if you want, we can discuss about that. So what is the message that I want to give you today? So first of all, mental navigation of conceptual relational structure, you can call cognitive maps, takes place across complementary allocentric and egocentric reference frames as during the navigation of physical space. And the internal exploration of conceptual space and possibly the construction of these mental relational schemas could be mediated in primates by attentional movements. Such attentional movements can be read out from spontaneous eye movements during the task, and their correlation with the task performance suggests a causal role and not epiphenomenal. Causal role that is supported also by strikingly similar results in macaques. So of course there is a lot more to do because we have to figure out the relationship between eye movement, attention, cognitive maps, attentional maps, brain activity, and so on and so forth. But we are doing that, and in particular, people in my lab are doing that. And if you're interested, you can listen to the Abstract Award talks tomorrow by Giuliano that is going to show you a little bit of what we are doing now, showing the neurophysiological mechanism of foraging in a conceptual semantic space. To conclude, I think that these results suggest that there is a continuity in the way we construct spatial relationship through our acting in the world that can be just locomotion, like going around somewhere. That's how we understand how things are related with each other spatially, or manipulation of objects. We understand how the relationship are looking around. And the same thing happened, and maybe that's the way we construct this relational, abstract relational mapping through attentional movements. So they are mediated by attentional movements. Okay, in doing that, I thank you for your attention. Let me thank my lab. Here you see some of it. There have been new people joining the lab lately. I would have liked to be able to acknowledge the work of all of them. I couldn't, but yeah, let me really thank them because they all this work would not be possible without them. In particular, Simone, Giuliano, Giulia, Yangwen, and Alex, of which I featured their work in this talk. My collaborators at the Max Planck Institute in Leipzig, so Stephanie and Christian, and the funding agencies that have been very generous in the last few years. And thanks to you again.
Thank you. That was fascinating. I want to get your thoughts on, I guess, maybe an assumption underlying how you're thinking about these conceptual spaces, which is that conceptual knowledge is best represented in metric terms, where you can then calculate things like Euclidean distance and so on. And you referred to early work from Roger Shepard, looking at that in color spaces. But at the same time he was doing that work, as you probably know, there was lots of work looking at non-perceptual spaces, looking at conceptual spaces, arguing that similarity was not well modeled in a metric space and led to the idea of ultrametric representations of like hierarchical tree structures of the sort that bears some resemblance to what Wilma was talking about in the second half of her talk. So I guess I'm wondering whether that matters for what you're doing. So when you get into conceptual spaces for things like animals that have then a hierarchical structure of mammals and birds, et cetera, and so on, whether or not you think this method is still going to hold up, whether it's kind of close enough, or whether you think there's a problem trying to apply metric spatial principles to conceptual similarity spaces that might not be metric.
Yes, that's a very interesting question. How can I start answering to that? Because it can lead to a long answer. But the short answer is that you can look at relationship, let's say, as a map or as a graph. Maybe a graph is much similar to the type of structure that you were referring to. So at the end of the day, at the moment, we don't know whether cognitive maps are Euclidean or not. Right? If you look at the early work from the Mosers on, that was the main claim. Right? We found the representation of Euclidean distance in Euclidean space in the brain. But actually, a lot of the latest model of the hippocampal entorhinal system, they treat this system more as a graph, as producing graph-like representation more than map-like representation. And of course, you can take a graph and make it a Euclidean graph, especially in some situation in which the space that you are exploring is homogeneous and symmetrical. And that is also the case in which, for instance, for grid cells, you find the grid. But as soon as you warp the space a little bit, you make it trapezoid, you make it irregular, you put boundaries in it, you put a reward or something, then this regularity disappears. Right? So that also suggests, and I suggested to many, that maybe these grid cells are not given a metric for space, but they're doing something else, although being involved in referring to spatial position. So I would say that I remain agnostic to whether which type of representation are actually hold there. Is that a Euclidean map? Is not a map, it's more like a graph. There are some models that can account for both. Right? For instance, the Tolman-Eichenbaum machine, which is a very important model for the hippocampal entorhinal system, can account both for Euclidean trajectory in a conceptual space defined by continuous dimension, but also for a family tree. And doing path integration in a family tree to discover this hierarchical representation. So it's difficult to me to give you an answer because everything is pretty much up in the air now when we go to that particular specific. We know, I think, that this region, these neurons are involved in structuring knowledge, but whether that is a Euclidean structure is not a Euclidean structure, when is one, when is the other, or that is it's really still a matter of hard debate and research. I don't know if I answer your question, but
Thank you so much.
Hi, thanks. This was really cool work. I was wondering two things. One, do you think or have you tested whether people are actually aware of this eye movement behavior, that they are categorizing stuff? And two, would you expect if you put constraints on how big of an eye movement they can make during a learning task, whether this would actually impair learning?
Yeah. So for the first question, no, they are not aware, at least as far as we know. We tell a cover story. We are looking at your eyes because we are interested in pupil size that reflect effort, cognitive effort. That is a plausible cover story, and people usually buy it. And then at the end, they don't say that they were realizing the people realize that they move their eyes when they think sometimes, but not necessarily accordingly or in the way that I show you here. So they are not aware. Yes, now a lot of future studies will be in my lab to assess the causality of this mechanism, as you say. One thing that we did is to ask people to keep fixation when they were doing the number generation task. So keeps on fixation there. And what happened is that we found the same effect. Like they were just doing micro movements, microseconds, but the effect was there and the statistical strength was exactly the same. So it's kind of they cannot really refrain for doing that. And in that part, in the case of the mental number line, this effect is very strong. Almost all the participants show that with 6 minutes of recording, basically. So yeah, we don't have data at the moment, but that's one of the next things to do is to look at causal mechanism. Yeah. Thank you.
There was a question here,
Yeah, thanks. Great talk. I was wondering about relationships of you being able to navigate spatially and you being able to navigate mentally, right? Is this like a null-sum game? Like if I'm bad at navigating spatially, I'm better at navigating my mental space? Or is it that those grid cells are basically, if you're better at navigating and making your space in real space, you're also able to do that mentally?
Yeah, I know only one study that tried to do that behaviorally without looking at the brain, and they found relationship between the two. But again, this is another, I think, portion of the field that is underexplored, and we should look into that. But yeah, there is an example that tells us that there is a relationship between these two things. But I can't think about any other further evidence for that. So I have to answer, I don't know. That.
Very nice, Roberto. I'm going to continue, go back to the talk, the question you just asked a few minutes ago by my friend and sometimes enemy when it comes to talk about concepts, and some of the discussions we had in Boston. I noticed that the space, semantic space you've been working with are pretty small spaces and low dimensionality. But your concepts have a much, much larger structure, and the question is, can you reduce it to 3 dimensions or small dimensionality to make it useful for navigational space? If not, then what is it you're doing? Mm-hmm. Are you doing not something about concepts as such, but rather the way of mapping a very large space of conceptual structure onto a particular model of relations that can be reduced to 3 dimensions?
Yes.
Which one are you proposing? Certainly, I don't think you're proposing that this captures conceptual structure in general.
No, no, definitely not. So that's also required, I think, kind of an articulated answer. No, to answer to your last question, no. And I think that the level of dimensionality here is important, or at least comes handy to explain this difference. The way large language models encode different concepts is through high-dimensional spaces, right? Many dimension, and this is necessary to capture all the semantic diversity that populate, let's say, or constitute our knowledge. And probably in the semantic networks, including the language network, different regions of the brain, there is this high-dimensional representation. Is there to some extent. And I don't think that that is what we are seeing here. Now, it's possible though, and that I think what is happening, is that when we are engaging in a goal-directed behavior, right? And when we, in order to do that, we need to take into account in a conscious way, maybe we engage in a mental simulation, or we activate like working memory. Anyway, we have to take into account a few dimensions of it. Then it's where this relational structure becomes handy. And also, for instance, when in extracting this low dimensionality in a particular semantic space, then maybe we can generalize to different semantic space. And this is an effortful act, cognitive effort. And that's where these low dimensional mental space are important.
So I understand you answering that, in fact, this is a strategy that we humans can use when we have very complex systems to perhaps reduce the dimensionality when we have to answer particular tasks, when we have to perform very limited tasks. But the same thing with the number line. The number line is a great idea, but 7.4312 is a number, and 7.4314 is a number, and the number line cannot tell you the difference. So the question is, there are two kinds of systems that we're talking about, and the attempts to reduce one onto the other is manifestly inadequate. At least it cannot answer the 2 points I've made. That's what I'm asking. What role then does these spatial kinds of representations play? And they are simply tools that we've developed to answer questions in experimental tasks.
Okay, no, I don't think it's just a strategy, although that is to some extent testable. But I think that, in principle, you have representations of knowledge at different level of dimensionality, and they're useful for different things. So, for instance, you cannot support like a conscious mental navigation in a high-dimensional space, right? You cannot probably consciously or mentally simulate more than 3 dimensions altogether. And that is a limit that we have, but that is also characteristics of an important part of our mental life, if you want, which is the declarative knowledge. At the same time, of course, you have millions of neurons that can represent stuff in high dimensionality and then do a lot of work behind the curtains, if you want, so that you can, that when I talk, for instance, I can choose the right syntactic structure. When I talk, I can choose the right synonym and say this word instead of the other word, and so on and so forth. But these are mechanisms I don't have access to. And probably they are high dimensional, or what happens, for instance, in the visual system, in the ventral stream, and so on. But when it comes to reasoning, when it comes to putting, for instance, mental analogy, for instance, that this low-dimensional structure becomes handy, because then you, when you have low dimensions, you don't have the course of dimensionality, and things becomes very clearly separated from each other. They become clustered. So then you can, you lose a lot of details, but then you gain in the possibility to generalize. And indeed, cognitive maps have been are involved in this generalization process, and that's what is thought is one of their features, if you want. So yeah, I think we are talking about two different systems there. You can make, if you want, but of course, so cognitive scientists are often said to reinvent the wheel, make it square, and call it a first approximation, right? So I don't know, I'm certainly doing a first approximation here, but hopefully I'm not reinventing the wheel. But it's similar to the two systems of thinking fast or slow, for instance, in that case. And some people have tried to make this parallel with the work on cognitive maps and what happens in other regions of the brain. I think that the direction where I want to go, or where I would like to go with this discussion, is that one.
Hi, great talk. I was wondering if you think that this mechanism could be the cause of learning disability. Usually learning disability use a map, external map, to correlate the concept or number. And so maybe, have you ever thought about this connection between learning disability and the categorization of concept, mental map?
Learning disabilities, no. I know that there is a strong, there is clear relationship between cognitive maps and disability like Alzheimer's disease, for instance, so spatial disorientation, memory disorientation, across this, from rodents to humans. And indeed, Alzheimer's starts in the entorhinal cortex, or at least some model of the Alzheimer's suggests that. But for learning disability, no, I cannot.
I'm not an expert, but I know that they use the external map, so someone that creates a map of concept to connect different concepts and to help learning. So I was wondering, I did just a connection.
You're right. Okay. See, yeah, it's true that at school they can teach you some conceptual map. Right? Is that related to this? I think so. There is a paper, a work that I just saw at the conference in which they show how this system is implicated, this hippocampal entorhinal system, is implicated and involved and important in creating the mental palace structure. You know, this strategy to keep things in mind and navigating and so on. So maybe this neural machinery would be important also for that type of conceptual map during learning that you are suggesting. But that's just a speculation. I'm not sure.
Okay,
yeah.
Thank you.
Thank you.
Thank you. I'm wondering about the stability of the gaze patterns that you presented. So you mentioned that they are idiosyncratic, so different for each participant. And I was wondering what happens, for example, if you were to bring these participants into the lab on another day. Would they look at the same positions for similar concepts, A, and B, if you were to add a third concept on another day, would they stay in a different position? Or so is it relative?
Yeah, it's interesting. We had the data for adding the third concept, but we haven't analyzed that yet, so I don't know. For the stability, they're less stable than what I thought. As I said before, I didn't show it. Let's see if I had it here. Can you see it? Yes, so if you partition the space, you would expect that in different blocks, right? If I look here for category A and then for category B, that is the same place that I do in the sixth run as I did in the first run. So we tested that, and actually we saw that that wasn't the case. There was this spatial drift over time of the experiments that changed with, in this case, the blocks, right? And if you visualize the gaze distance matrix here that is taken across all the participants, and then you do multidimensional scaling, so a reduction of dimensionality, you see that the gaze is separating the two categories, but it's also varying a lot in time. So from block 1 to block 8, basically people are looking at a very different position in the screen. But keeping the two categories separately. So these gaze dynamics are picking up to some extent. We don't know whether that's important or not, but also the time that is passing by during the experiment. But when, for instance, we look at the correlation with the performance, the temporal drifts did not correlate with the performance at all, whether some subject have a strong or a weak temporal drift. But the category separation, yes, was important to retrieve more words.
Can I ask a follow-up
Sure.
question? So a thought experiment, if you were to immobilize the eye muscles, would they become worse in this task?
Immobilize the eye muscles.
Yeah. By some
Is that ethical? Can we do that?
I called it a thought experiment.
No. I was asking for maybe there is some transient. Yeah, that's a very good question. It's about the causal role of all of this, and that's something that we need to investigate, as I was saying in one of the previous questions. There are the way that we were thinking about was just to direct like the gaze in some particular way and see whether that people make that people think in a particular way. That's to say it in a very generally, or maybe restrict eye movement to some extent with fixation. But the idea is yes, there should be an effect on the performance.
Thank you.
Thank you. Whether it is enough or not is a very hard question to answer. I don't know. I don't have a metric or an example to which I can compare and understand. For the time course, yeah, we looked at that. It's longer than 500 milliseconds. For the number effect is sustained for all the 500 milliseconds and even before. For the color effect, which is weaker, it peaks at 300 milliseconds before they are saying the word. So, but that's something that we definitely need to replicate and to see whether that is the case also in other conditions. But in principle, saccades are very fast, right? And also we don't know yet exactly what is the precise oculomotor behavior that is associated with that. Is that a saccade? Or is that some smooth movement, like around that create this effect? Because admittedly, we are looking at that at the moment in a very coarse way. Just we take 500 milliseconds, take the median XY, but in there, the eyes move a lot. We are trying to do better analysis with scan path now and more fine-grained analysis, looking at the saccades, see whether or not a person makes saccades in that 500 milliseconds is important for the effect or not. Yeah, because in principle, I don't see that the ocular effect should be necessarily long and sustained in time to be causal, right? Maybe not.
Sorry, Brad triggered this question. So the example categories that, if I recall correctly, were animals and professions?
Yes, exactly.
Now, these two categories, I don't know how many others you used, but they seem to differ in one critical aspect, which is the imaginability, right? So we know that participants tend to move their eyes when they imagine stuff. So can you rule out that differences are due to the fact that one of these categories is rather concrete, so you can imagine animals when you recall them, whereas a profession is much more abstract, and so therefore you find gaze patterns because for the concrete category there's clustering which you don't find for the more abstract category? I know this is maybe far-stretched, but...
Yeah, I guess we can test for that, like looking whether there is more or less dispersion, for instance, for one category or the other. It doesn't look so, but yeah, I think it's an interesting further control to run. So yeah, it's a good idea. We're gonna look into that. Yeah. Thank you.